I was a little fascinated about the how the epithelial cell in the gut can do two jobs. So one is blocking all these, external bacteria, pathogens, antigens, while still selectively absorbing the nutrients.
Welcome, everybody, to the Poultry Podcast Show. My name is Doug Korver, and I'm one of the Poultry Podcast hosts. Today, we're very happy to welcome Dr. Yihang Li from the University of Delaware, Department of Animal and Food Sciences. So welcome to the program.
Okay, thank you. Yeah, hello, everyone. I'm Yihang. Yep.
Yeah, so can you tell us a little bit about yourself? What was your path to your current position?
Oh, yeah. I originally from China, and I graduated from China Agricultural University, where I studied the monogastric nutrition. And then I came to the US and I landed at North Carolina State University. I studied the animal nutrition as well, but more focused on the baby pig calcium nutrition. And after graduation, I joined to the Michigan State University for my postdoc training. There, I get exposed to the gut physiology and gut health, and I'm really passionate about that. And I got a couple of publications. And at 2018, I started my faculty position at University of Delaware, focused on the animal nutrition, gut physiology, and so far it's almost 5 years. So, yep. That's
Oh,
about
that's
me.
great. So you mentioned that it was in North Carolina where you first got interested in gastrointestinal health research in pigs. But what was it about the GI health research that really intrigued you or interested you?
Oh yeah, I was a little fascinated about the how the epithelial cell in the gut can do two jobs. So one is blocking all these, external bacteria, pathogens, antigens, while still selectively absorbing the nutrients. And as my background training in animal nutrition, we care all about how the nutrient can be digested, absorbed, improve the growth performance, the blood biomarkers. But we really didn't, stop and look at how this process is actually working. And once I was training in gut physiology lab, I got a chance to expose to the Ussing chamber. It's an electrophysiological instrument where I can put the fresh as alive gut samples on the chamber, and I can see how am I adding the nutrients, the gut start to absorb the nutrients. That makes me really excited. And
Mhm.
and meanwhile, we have some training about the nutrition and the immunology. So there, I was studying one of the immune cells, the mast cells, and I clearly observed how the nutrition and the stress can directly affect the gut repair themselves, activate the immune systems, and totally reflect to the performance. So as you know, like once we study the performance, it's like a black box. We just see the animal change, but we don't know what is the reason. But here with the gut, with the real tissue exposed to you, and you can dissect the reasons very clear. That makes me very interested. So I'm continuing this work. Yeah.
Yeah, that is fascinating.
Eastman serves veterinarians and nutritionists in agrochemical and animal health industries by helping them select, evaluate, and implement innovative nutritional programs. Eastman works with your team to customize your gut health approach in feed and water. Eastman's approach addresses nutritional and bacterial challenges and finds new ingredient preservation and hygiene solutions. Explore ways to accelerate and innovate your programs. Contact the animal nutrition team at eastman.com.
So in China, did you work with swine primarily, or did you work with poultry as well?
Yeah, I got the opportunity to study in I think it's still now is the top lab in China, study monogastric nutrition. We have both opportunities. I worked with the pig a lot, but my master training is based on the digestive enzymes, the NSP enzyme, how that help the animal release more of the energy through the gut. So I was working with the whole process from the gene editing, fermentation, and purify of the enzyme and feed to the broilers. So I have a lot of experience with the broilers nutrition and feedings. But yeah, both pigs and chickens, I review my background of training about 20 years so far, half and half. So half time I
Yeah.
work with pigs, half time I work with chickens.
Okay. Do you think that working in pigs and chickens, what you learn in one species kind of helps you understand what's going on in the other?
Yeah, definitely. I started with the pig, and I was lucky because the pig is relatively bigger than the chicken. So when you go to the dissection, the lab process is easier to see, is easier to manage. With the chicken is tinier, and now I'm working on something with the embryo development. So how the intestine develop itself from the beginning, from the stem cells. And well, the embryonic tissue is so tiny because I have so many years of experience, so that's not a big issue to me. And this I can see more broad applications. So with the pig, we know that's the large animal as a model animal. So we can expand research not only for agricultural, but also basic science and the human biomedical research. And with chickens, now I'm staying in the University of Delaware, and Delaware is a big chicken poultry state. So we have a lot of applications directly to the agricultural for sure. But as I said, I'm working with embryonic development now, so this model give me a better access to the embryo itself without the maternal, like from the mother, from the milk, from nutrition. So it's pure model. So I think both sides serve with the science work better and have multiple applications, not for agriculture alone.
So, can you just summarize for our audience why you think gut health is so important in poultry production?
Yeah, that's a very good question. I think now everyone knows that the restrict use of anti growth promoting antibiotics, biotics. So that's a concern. What can we do, right, without adding these things to still maintain the health gut? And we know the gut is a major organ for absorbing nutrients, to blocking the pathogens. It is the biggest organ, has the biggest surface area compared with skin. It's larger than the skin. And how efficient. Now we're not only looking at how big the animal can grow, but also how efficient the animal grow. And the gut health is the key. I think is the key contributing to the efficiency and also the animal health. So I think we talk a little bit about this endocrine function with the nutrition and the gut function microbiomes. So I think it could be the future that we can understand first and maybe manipulate the gut feelings and transfer these feelings to the animal's hormone level and anti-stress.
Mhm.
So yeah, so gut definitely is very important to me, the most important organ for this poultry.
Right. You're using some really interesting techniques in your lab. So, can you talk about enteroids? What are enteroids?
Yep, yeah, that's a good one. I have this questions most time. So Enteroids is a newly developed idea for the in vitro understanding of the organ's function. So at the beginning, we all know we can isolate the cells and make it a cell line and study the cell function in the past. But this is just mono cells. You only have one pure cell population. But as the technology developed, nowadays we can culture multiple different cell types in the same culture, and then they isolate from the gut or from any organs as the stem cells. So it's naturally developed without the animal's body support. So in a petri dish, you can develop an organ. So this is a mini gut. We can consider it's a mini gut that has three-dimensional structure. It's better than just the epithelium cell, like a monolayers. It
Mhm.
has multicellular component. It has epithelial cells, immune cells, some nerve cells or muscle cells. So in the physiological function, it's more close compared with the monolayer cell model, more close to the body, like in vivo physiology. And we can measure a lot of functions that we can never done. So there are two types of the enteroids in terms of the gut. So one is the very basic, like scientific research related, is originated from the stem cell, and its culture, it can propagate. It can, once you have this successfully cultured, you can have this forever. And another type is what I'm working on now, is initially developed from the Roslin Institute in Scotland, and we adapt their protocols, and we can isolate embryonic gut, and then just kind of have a snapshot of that gut embryo, and we have the benefit of the apical out structure. Means if we add something in the culture, it just mimic what we add orally into the chicken, so the gut can sense this molecules or nutrients. With the traditional enteroids, it's inside out, so all the lumen side is inside. So it's good for research, but not so good for trying to high throughput screening for product. So with the both model, we can go one side, study the basic stem cell function, go the other side, can screen nutritional or orally developed delivered molecules. Yep. Okay. So that's general enteroids.
Right. So it's not just taking gut tissue and grinding it up and basically looking at little chunks. These are actually developed.
Yes.
So the cell types, you mentioned there's different cell types present in these organoids. Is it fairly reflective of the composition of the gut tissue, or does it skew towards epithelial cells or enterocytes versus immune or what have you?
Right. So that's another difference between these two enteroids. The enteroids we work on with the embryonic tissues, that covers everything you can see from the real gut. So it has the top layer of epithelium cells, and within that layers you have multiple different cell populations, starting from the stem cell all the way differentiate to the goblet cells, to the penny cells, to the top cells, or the enterocytes. And each cells, they're producing their matured protein profile, so they're real functional cells. You can detect the mucin, defensins from the penny cells, and all these nutrient transporters, digestive enzymes, everything. Versus another enteroids I mentioned, you isolate from the stem cell, and that's luminal inside. This type of enteroids only have the epithelial cell, and it has the properties that you can propagate. It has a stem cell. You can always have the new, fresh gut epithelium grow, but you're missing the important immune cells beneath the epithelium. Versus the embryonic enteroids, beneath the epithelial cells, it has some leukocyte, some lymphocyte, some nerve cells. So all the reactions from not only epithelium but beneath, you can detect in the lab.
That sounds like a really powerful research tool.
Oh yeah, that's very, very helpful for
Yeah. So you also talked about Ussing chambers to study intestinal function. Can you describe what they are and how you use those to measure gut function?
Okay. Yes. It's quite interesting because I'm originally from China. I learned everything English from here, but some people pronounce it like Ussing chamber, sometimes pronounce the Ussing chamber. I would just go for that. So I learned it from my postdoc lab, and that's another very, very powerful tool to study real gut function. So here you are looking at a real tissue. It's a gut tissue. So it requires a fresh gut. So immediately after euthanize of the animal, you have to take the gut and use this Ussing chamber. It's not like other technology. You can freeze the tissue and deliver to a different place and wait for a couple months. This is not. It's real, it's fresh. And that means it has a fresh physiological functions. So once you put this on the chamber, it has two pieces of chambers, and your gut tissue is in between these two chambers. And the chamber is filled with a Ringer solution, which has the necessary ions like sodium, chloride, potassium, to make sure the gut is healthy. And on outside this chamber, you have the water bath to maintain the good temperature. Within the chamber, you give the oxygen tubing to keep bobbing into the oxygen, support the tissue's viability. And you can also insert the electrodes close to the tissue where you can measure the current and the potential and the resistance of the tissue. So we know all these cells have they are polarized, the tissue, so you can detect a different electric potentials, and that reflect the function. So for example, if a healthy tissue, the basolateral side of the cell can keep pumping out the sodium and pumping in potassium. This generate a potential change. And the higher potential means that the tight junction is tighter, the tissue is more integrated. So then we can use this to reflect the barrier function of the tissue. So say if you're challenging one animal, or you're feeding, say, butyrate or some beneficial nutrients, and then for a while you isolate these tissues, put on the Ussing chamber, you can clearly see how tiny but very significant difference between these treatment on the gut barrier function. And also, we can add some nutrients, say glucose, individual amino acid, or peptide. So we add these to the luminal side of the tissue. Because it's live tissue, their job is absorbing nutrients through transporters. And this activity generates current. And you can clearly see the current measurement was a flat line until you add the nutrients. Immediately it increased to a peak. And this difference reflecting how strong, how quick the tissue is absorbing nutrients. And you can easily make comparison. This some type of thing you can never see in a real animal level. You have to take the gut level. So that's the nutrient absorption. And you can also have the labeled, say, molecules, phytidextrin. So that's a molecule we always know. We can do the oral testing and measure the blood, how good the gut is, leakier or not. But the problem with the whole body is like you're measuring all the GI tract from the esophagus, crops, the jejunum, ileum, cecum, everything. But this Ussing chamber, you can specifically dissect. If your product is working on jejunum only, you only isolate the jejunum and measure. And believe me, these different segments of the gut definitely do different jobs. And if you measure the overall effect, you may, underestimate certain effect. So this is very powerful to study specific gut function, permeability, leaky gut, nutrient absorption. And in our lab, we recently developed a way to study the endocrine function. So the gut have the endocrine cells. It sense the microbiome. It sense microbiome metabolites or the nutrients. And different profile there will activate different receptors in the cell, and then it secretes the hormones to the serosal side of the chamber, so we can measure very clearly how much the hormones change, the concentration change in the serosal chamber. So we have done this in the stressed animal and also nutritional treated animals, and we clearly measured some hormones, for example, the peptide YY, GLP, the glucagon-like peptide. Those peptides, their function is regulating the body glucose, and they can control how much the glucose absorbed. So the gut is a very fascinating tissue. Like it senses the environment and decide if there's so much nutrient, it upregulate absorption. If it's less, it's downregulate to reduce the maintenance. So it's all correlated to the luminal treatment. So how much the hormone secretion we can measure. So this is another strong power of the Ussing chamber. The very last one, sorry, it's long.
No, that's great.
So into this technique. Very last one I want to tell you is the gut nerve function. So we know the enteric nerve system is very big. It's almost your second brain. They say there's so much neurons it cannot fit in the brain, so it's put in the gut. Right. So a lot of things has been done in the human-related field, but in agriculture, very, very minimal, especially in the poultry. But with using chamber technique, you can detect the nerve activities. We have done this in our lab. So from the human literature, we know some drugs can activate the nerves. So it can fire certain neurons, and these neurons will push the signal to the epithelial cells to increase the secretion. So that's one of the mechanisms you have when you have diarrhea, right? So activate nerves, activate chloride secretion, and you have diarrhea. So with these drugs, we can measure how much the sensitivity of the nerves. So if certain treatment, in our case, early life stress, if they're a stressed animal and then you activate the nerves with the same dose of the drug, the secretion extension is very big. So it's more secreted. So they are hyperactive nerves. You cannot see this with any other technique, but using chamber can do. And also you can block certain nerves. Say you have clear cholinergic, use acetylcholine to activate, or use some other neurotransmitter. So you can block these receptors and really study what nerve there is controlling the secretion function, absorptive function. So the applications can be understanding the nerves. Maybe in the later someday, we can develop certain specific drugs that can manipulate the gut functions through the nerve functions.
Mm-hmm.
Yeah. So yeah, with all these nutrient absorption, barrier function, secretion, endocrine, and the nerve function, we can all do with the using chamber.
Right. Wow. So let's follow up on that. You recently co-authored a review article on intestinal enteroendocrine cells in neurological and psychological disorders in humans. Would you expect to see a similar relationship in animals, or that is, could we help animals deal with stressful situations like vaccination or transport or what have you
Yeah,
by modulating the microbiota or modulating gut function?
Yeah, I definitely believe so. As we all know this, recently, like a microbiome is very well studied, and still we have blanks, but more we understand than before. And in the human world or model animal world, we see your mood can be changed by the microbiome, right? Your metabolism can be changed by microbiome. I believe that's true for all these animals, vertebrate. So in the chicken, I believe the same. So when the microbiome get changed, their metabolism pattern get changed, the gut will definitely sense that, right? So the sensing is very important. And after sensing, the signal will pass to the functional cells. Say endocrine cells could be the first one. Endocrine cells send some signal and pass to the nerves. That could be a second one. And then the nerves will fire the different things through the brain gut axis or directly in the gut, say motility, the gut muscle. So definitely, I believe different um, metabolites in the microbiome in the gut can control this hormonal level. And in our study, it's not published yet, but we have observed the level of these hormone get changed, and we are trying to correlate with the microbiome change. So we were able to isolate certain gut content and then sequencing and then predict the function of this microbiome and then link with the endocrine function. So I believe there's a strong correlation there, and it could be the future. So one of the direct application you can imagine that the I'm not sure if you have heard the term of ileum break. So that means if your ileum have sensed certain level of nutrient, it just like a press a brake, so it slow down your GI digestion, it slow down your feed intake, so you don't overwhelm, you don't give extra burden to the GI tract. So one of the mechanism is the CCK and the peptide YY. So these two hormones secreted by the gut and reflect to your brain. And also, you may know this serotonin. Serotonin is a hormone make you feel calm down and you feel happy. And 99% of serotonin is produced by the chromaffin cells in the gut. So these cells definitely will sense whatever happens in the gut and then pass signal to the brain. And in our study, we find the feed intake could be regulated by these. So like I said, the ileum brake, if the animal eat more and they have more of these hormone produced, and then it kind of slow down the appetite, right? So in
Right.
our study, we find the stressed animal missing this process. So they have less activated endocrine cells. So they produce less of these stop hormones and make them eat more. And then that's very interesting. You see, stressed animal, once they are stressed once, they always eat more. And
Hmm.
they always want something to satisfy themselves to feel calm. So we believe this could be one of the mechanism.
Okay. So does it work the other way as well? Because some of your research is involved or has looked at the effects of delayed access to feed. So in broiler chicks, placement in the barn and access to feed. So
Right.
is there a role of some of the interactions you've talked about? Um, does that influence the response to delayed access to feed?
Could be. Once the animal are delayed in our model is kind of delayed access to the feed. So that's one thing, they are stressed, and another thing, they are malnutrition. So they don't have enough energy and protein to support. At that time, I think the animal try to survive. So they are evolutionary speaking. They have all this machinery prone to the mechanism how can make them survive better. So I think this could be one of the mechanism. They have upregulated glucose absorption, and they have reduced endocrine function. So these two together make them eat more. And we have seen, although the gut function still have a long-term difference, but the body weight, they try to catch up. There's a catch-up growth mechanism. Although we don't know what is pure mechanism for catch-up growth, but I think the change of the gut absorption, digestion, appetite control can be the mechanism for catch-up growth. So they just need eat more than they should, or they can, to catch up the delayed couple of days of the feeding. But yeah, for that case, we have seen the gut barrier function was damaged because of delay, like delay feeding or stress. So long termly, say we only delay the animal for 3 days, but we keep the animal for day 42. So long termly, we keep measuring the gut barrier function. They always have reduced the barrier function. So their gut always stickier. But at the other side, their nutrient absorptive function get increased and their hormone level reduced, so that makes them eat more. So we believe if it's a good environment, this stress make them survival better. But if it's a bad or not optimal environment, say you have poor biosecurity or a lot of pathogens, they are less powerful to deal with the infection.
So it really is important if there's a delay in feeding to make sure that those chicks have a really good environment.
Yes, yes, yes. And then they don't lose too much of the performance.
Right.
Yep.
So in your research on gut health and some of these really intriguing interactions between different systems in the body, are you looking at particular feed additives or nutrients and how to maybe manipulate the gut into doing the things that we want it to do?
Yeah. That's as a goal. We always a little more greedy. See, now the breeding is already so good that the birds can grow so fast, but we still see what else we can do, right? Make them more healthier and something. So yeah, nutrition is a good way. We all understand, like the cheaper way to improve functions. So we have tried additives like essential oils, like everyone look at this like organic acid, essential oils, or the combinations, and we see how that help the animal with the antimicrobial, anti-inflammation, antioxidant functions, and everyone studied this at now. But we we have the advantage of using this Ussing chamber and Enteroids model, so we can do a quick high throughput screening on different combination and the dose. So if you go to the literature, we all know butyrate is good, we all know curcumin is good, but what's the blend? What's the composition of these and work as a whole, and the optimal dose? You see controversial data and the different so many different things, with or without the stress. So sometimes the data looks good, but once you have the challenge, LPS challenge, it's different. So with our model, I think we can closely look at the physiology of the whole tissue and then the true epithelium, like the enteroids. So we can clearly understand which part activate the immune system, which part benefit the barrier function, and the combination. So that's one thing. And another thing I forgot to mention to you, but I studied early life factors a lot. So in my PhD, we studied the early nutrition in the pigs, so before they're weaning, and now and also the early weaning for the pigs, and all the early life. And now in the poultry world, I study the early nutrition. So I gave the eggs the in ovo nutrition, so we inject certain nutrients there, and study how gut develop changed and the long-term benefits after hatch. So we apply this Ussing chamber, we apply this Enteroids technique in there. We have tried the glutamine, although it's people know about this, but still with the new model, we try to see if we can understand new things. So we in ovo inject the glutamine and see how that support the intestinal epithelial cell, epithelial stem cell, and the endocrine cells development. So yeah, we have some data there that's quite interesting. And now we are moving to the next step. So with the in ovo nutrition plus the early life stress, and see if the combination makes the gut different.
Yeah, if the early nutrition helps to protect the birds,
yes,
through that stressful period.
Right. And if we understand this aspect a little more, we can probably selectively pick drugs or nutrients that can, we have a target, right? Endocrine cells.
Mm-hmm.
Which one targets the endocrine cells the better? So the chicken always have good advanced feed intake, or they feel calm, they're stress resistant. So that could be the future. So use the early nutrition manipulation on the endocrine function development, and then for the post-hatch gut health. Yeah, that's something we are working now.
Yeah, that's great. So just going back to the discussion on really alternatives to antibiotics. It was really interesting. I thought that you're able to screen combinations and dosages in vitro before you try in vivo, because one of the things when I talk about alternatives to antibiotics is there's no magic bullet. There's no one thing that, we take out antibiotics and we can put this one thing in and it solves all the problems. And it's just such a complicated situation out in the real world where different combinations are probably going to be required in different circumstances. So I'm really excited to hear about your research and the ability to screen under different circumstances, different combinations and dosages. I think that's really interesting work.
Cool. Yeah. That's good. Like we are still beginning. I cannot have a a good answer for anything, but we that's the direction we are working on. Just like you mentioned, different component have their strengths or weakness. So some component really beneficial for the epithelial cells that upregulate the tight junction proteins and make the gut stronger. But then at the meanwhile, it's less turnover, so the gut lives longer. And then it may not be perfect if you have certain things change, like you change the feed or change the microbiome. They are less active in deal with the change, adapt to the change. Some component really good at anti-inflammation, and we can detect this the whole cascade of the inflammatory pathways, and then we can analyze these components function. Some really good at reducing or increase the antioxidant enzymes. So these different ones work in the different angles, and the gut is a whole tissue, and the dose you use is it can be different, like how much you have it here in the culture versus how much you feed the animal. So that's one thing. And also another thing is, if you have, in the traditional way, we have the epithelial cells. We only care about this epithelial layer. And we think, okay, butyrate works really well for this, for example, butyrate. And the higher dose makes it better. So you have a range and you detect, okay, this is best. You should feed in the animal. And then if you put the immune component inside, so you have the epithelium covered with the immune cells and you still use the same level of butyrate. And the issue is the extra butyrate can be toxic for the beneath immune cells, right? So then it makes things flipped. The more you add, the worse. So with the current Enteroids model, we can put all things together. We can think, okay, maybe the dose is a little low than the other, but that's the happy medium. So that they didn't irritate the immune system, but also maintain the barrier function. So things like that, I know it's old story. Everyone talk about butyrate, but still we have rooms for refine things.
Yeah, and I think that's maybe been a limitation in the past of, we see these interesting results in the lab, in vitro. We try and translate that into small scale research studies with live birds. And then we have to bring that out to industry where, we're dealing with millions or billions of broiler chickens. And it's not always a smooth path. And I think, the work that you're doing really is an important step in between that benchtop discovery and even the small scale research. So, it sounds like a really powerful approach to streamlining that transition from ideas and discovery to practical application in the industry.
Yeah, that's my passion. So I also, I have a background of nutrition. I know animals, and we are so focused on the performance, FCR, ADFG, ADFI, ADG. Once I got trained in the vet school for my postdoc, people look at different angles. They look at the immunes, they look at the physiologies, and each field have their strengths. So my goal is to translate in the other way what we can learn from the human or biomedical research and see what new things we can apply to agriculture. It's a little opposite with learning from animal models, but we can apply new knowledge to the benefit the agricultural field.
Right.
Yeah, that's my passion.
Yeah, great. Are there any other current research projects you've got going on that we haven't talked about that you're really excited about or you want to mention?
Yeah, that's the one. I think we covered most. I gave all the things I know. Yeah, I also work with pig side, but I don't think it's fit here for poultry podcast. Yeah, I think now very interesting thing. Another I will try, but we haven't talked about. It could be the biomarkers development. So, when you talk about gut health, you have a fancy physiological instrument, you have cell culture, everything, but in the farm, you cannot do this, right? You just want to take saliva samples, feces samples, or blood samples and know things. So that could be something in the future. We are also working on this, but very slow. The correlation. So, say you can do metabolomics, you can do RNA sequencing, you can do the 16S microbiome sequencing, metagenomics, everything. So, how to correlate these together could be an interesting story, and how to use the real gut function as your criteria. So okay, you want to treat this animal with stress, you want to treat this animal with challenge or nutrition, but the gut responses may not be the same. So in your treated group, they may resistant to stress. They may not respond. They may not be sensitive to treatment. So what you really need to do is to measure their function, and then sort these animals, and then you have a very good populations, and then you can do this correlation thing. But most of the lab or the works now, it has a limitation. People don't have resources or money to really understand the gut functions. So I think it will be beneficial if someone know about the Ussing chamber, about the enteroids. They can fully identify the gut function, and then use this as the standard answer and to correlate with the modern sequencing, next generation sequencing technique. That could be cool.
Natural Biologics is using cutting-edge science to dig deeper into the poultry health challenges you face. By gathering scientific evidence, they identify the most effective combinations of natural ingredients that improve animal health. Visit naturalbiologics.com/poultry to see the newest research in both turkeys and chickens.
Yeah, and I think it goes back to something you said earlier in our interview, is that our outcome is really growth and efficiency, and those are pretty easy to measure. But understanding all the things that contribute and the complexity of systems within the body that interact with each other and sometimes counteract each other, it's just really exciting to hear about the research that you're doing and the application. So. Is there anything that we haven't talked about outside of research that you want to mention, or this has been a great conversation? So
I like this, and I appreciate this opportunity, and I like to talk to you. you understand everything here, and very encouraging. So that's good. I think that's pretty much what I have been trained, what I'm doing now, what my passion is, and how we can help with this the both understanding of the science and then in agriculture. So that's the beauty of working with animal science. You study biology, you study physiology, but you have a direct contribution to the field. So I think that's good. Any collaborations, working with people, that's
I think your passion for your research really comes through, and so it's been great talking to you. Thank you, Dr. Yihang Li from the University of Delaware. It's been really enjoyable for me, and I hope for our audience. Thank you to our audience for joining us, and I look forward to future interviews maybe down the line. And I hope our audience enjoyed this interview.
Thank you.
Thank you.